third and final speaker for this session is Dr. Bruce Beutler, who is the Director for Center for Genetics of Host Defense and Professor, the Raymond Ellen Willey Distinguished Chair in Cancer Research at UT Southwestern. Dr. Beutler did his undergraduate work at UC San Diego, followed on by an MD degree at Chicago in 1981. He then went on to UT Southwestern for his residency, spent a little bit of time as a postdoctoral fellow and assistant professor at Rockefeller, at which time he had isolated the mouse tumor necrosis factor, TNF, and then in 1986 went on to become an investigator of Howard Hughes Medical Institute. During his research, he's worked on designing recombinant inhibitors of TNF, which are widely used to treat rheumatoid arthritis and other inflammatory diseases, and then later on studying the function of toll-like receptor 4. In 2011, he was provided with the Nobel Prize in Physiology and Medicine for his discoveries concerning the activation of innate immunity. Please welcome Dr. Beutler.
Well, thanks very much. It's a real pleasure to be here. I always have to be grateful to JAX because many years ago, in 1965, it was discovered that a strain of mice that they preserved since separation from the parent C3H strain, C3H-HEJ, was LPS resistant. And I spent many years positionally cloning the mutation in these mice in order to find the endotoxin receptor. And that eventually led to a Nobel Prize for me. So I hope among the 26 that you count, that one is one of them. I'm not really sure. We know today, of course, that mutations in this gene occur also in humans. We know that the whole system is very important for mounting an innate response to microbial infections of all kinds. And this was a very important mutation to find. It was an example of the classical genetics that begins with phenotype, in which there are alternative states of being for an organism, and one can ultimately explain them at a molecular level. I'd argue that much of what we know about biology arose from studying spontaneous mutations. But unfortunately, they're very rare, and they're most often ignored because their effects aren't particularly obvious. One has to screen for them. In certain instances, one has to have a particular interest in order to find them. As you all know, it's possible to plan for discovery and to create mutations at random, generating phenotypes of interest rather than simply waiting for them to occur. And that's the essence of forward genetics. It's making phenotypes at random, then solving the phenotypes, finding the mutations that are causative, and then one knows something that one didn't know before. This is a tremendously powerful technique because, first of all, it's unbiased. Second, because there are no guesses made about the system, it yields genuine surprises, and one comes away with knowledge all by creating exceptions to the norm and then explaining them without guessing about how the system works. On the negative side, historically at least, forward genetics was expensive and very slow in mammals. It took us 5 years to track down the LPS locus, for example. Nonetheless, I loved this technique, and I pushed forward with it, confident that better times were coming. And there were a number of advances, of course. The first probably was the sequencing and annotation of the mouse genome, after which one never had to make a contig again or explore it for genes. Then there was the development of massively parallel sequencing platforms, and finally the development of exome sequencing for the mouse, which sped things up even faster. But with each of these advances, a new bottleneck was exposed, and I'll come to that and how we've overcome it and how it's now possible for us to identify the causative mutations in real time. We've been using ENU as a mutagen in mice to create phenotype for about 14 years. And ENU is the only chemical mutagen that can be used in the mouse. It creates point mutations, And in our very extensive experience and in the experience of the whole field, when you see a phenotype, it almost always emanates from a change in coding sense. And this, of course, restricts the search area that one has to pay attention to. In Dallas, over the last 3 years or so, we've archived 123,878 G1 mutations from mice born to mutagenized fathers. And these changed the coding sense of 19,624 genes, which is 86% of all the genes that code for proteins in the mouse. Of these mutations, 9,942 create probable null alleles in 6,361 genes, so we've overtly inactivated about 28% of all genes in the mouse. But we have to remember that many missense alleles also cause damage or cause null effects, and we believe that we've probably mutated about 50% of all genes to phenovariants. This is not to say that all of these were studied for phenotype in an organized manner that I'm going to describe to you. 3 years ago, we began to realize that the new bottleneck was in genetic mapping. We mutagenized, we screened, and we found that we could declare phenotypes, but the number of phenotypes quickly began to exceed the number of mutations that could be assigned causation. Our standard procedure in those days was to take a mutant mouse, generate a homozygous stock in most cases, outcross it to another strain, backcross, and then phenotype the F2 mice in order to map mutations. And that had become too ponderous given the rate of production of mutations we could achieve. Also, it wasn't lost on us that we were ignoring most of the mutations. In the classical approach, one would simply look for one mutation, the causative mutation, and all other mutations were ignored. Therefore, one didn't know how much damage had been done to the genome, and it was impossible to exclude or exonerate genes from their role in a phenotype of interest. I would say that the long-cherished dream of all positional cloners was to skip all of this and to have a magical kind of glasses that one could put on so that if you had a phenotype that was obvious like this one, or even one that wasn't obvious, one would be able to look at a pedigree of mice and divine agency would point out to you which mice were affected. And not only that, but within the blink of an eye, it would tell you this is a mutation in SOX10. These are the coordinates of the mutation. This is the amino acid change. This is the motif that's affected, and perhaps it would even give you structural information if such were available. And what I can tell you now is that this approach is really something that we use every day. And as far as I know, we're the only ones to use this approach. But at present in our laboratory, when a phenotype is recorded, the cause is known in approximately 1 hour, or the phenotype is rejected as non-genetic. And this applies to all phenotypes that we study, whether they're visible or immunological, whether they're qualitative or quantitative phenotypes. And the latter are better to study, in fact. Cost is independent of the number of phenotypes that come from a single pedigree. We don't need to have 100% penetrance of the phenotype to track down the causative mutation unambiguously. And our approach lets us measure actually how much saturation has been achieved in the course of the whole mutagenesis exercise rather than to make probabilistic estimates as we used to do. It also lets us exonerate genes that are not involved in the process of interest. How do we do this? Well, first of all, we make G1 mice, as we always have, and we try increasingly to make G1 mice that have contributions of mutations from 2 G0 fathers that are both mutagenized with ENU, and in that way we increase the concentration of mutations in the G1. All G1 mice are routinely whole exome sequenced in order to find all the mutations that affect coding sense that they might transmit to a G2 or G3 mouse. From fairly long experience now, we've looked at 1,197 G1 mice made by the old-fashioned pedigree construction method, where we cross a G0 simply to a B6, we know that every sperm carried by a G0 mouse that transmitted to the next generation has an average of 63 mutations with a modal number of 70. And if we make these new pedigrees with the incorporation of 2 G0 mice, then the number transmitted to the G1 goes up to 93, which is about what you'd expect. We now do some selection, and if there are fewer than 30 coding changes present in a G1 mouse, we discard the animal. But if there are greater than 30, we move on and we order an AmpliSeq panel, which permits us to genotype all of the mutations identified in the G1 in its descendants, and to do so in a single reaction for every mouse, be there 100 or even more mutations. It doesn't really matter. At this point, the G1 mice are bred to make G2 animals, and typically 10 daughters are produced per G1 by harem breeding. Those daughters are back-crossed to the G1, and we aim to make 50 G3 animals of both sexes for phenotypic analysis. Prior to the distribution of the pedigree for screening, all of the mutations are genotyped, so we know the zygosity of every mutation in every G3 mouse, also in the G0 prime if it exists, also in the G1. We've been at this since about April of this year, and this tracks our progress in terms of validated mutations that have been genotyped in all of the descendants of G1 mice. We've now banked approximately 37,000 mutations, and if we look at the number of alleles, since some mutations are transmitted from the same G0 into multiple pedigrees, we have slightly a smaller number, and if we look at the number of genes, we're closing in on 15,000 genes having been modified in some way. So this changed. We know exactly what damage has been done to the genome. So one may think of this as having just dumped about 37,000 mutations into the coding region of the genome, which of course is quite a lot of damage. And then all of the G3 mice are subjected to a screening pipeline which begins with visual inspection, then measurement of body weight, a glucose tolerance test, and then a large number of immunological assays. We test both innate immune competence and adaptive immune competence by immunizing the mice. We look at their ability to repair the damage caused by dextran sodium sulfate. We look at their ability to survive challenge with a viral pathogen, mouse cytomegalovirus, and the titer of virus that accumulates in the spleen. We also do flow cytometry both before and after that screen. And then the mice, in some cases, are passed on to neurobiologists at UT Southwestern, Joe Takahashi and Qinghua Liu, who look at a screen for innate fear based on olfactory cues and also for perturbations affecting circadian rhythms. And of course, we could do much more. This is one of the limiting factors in the whole process. To find the mutations instantly, we rely on the principle that one doesn't need to do an outcross if one is confident that all the causative mutations have been identified. And instead, one can simply look at the pedigree directly. The sequencing and genotyping are done in advance of screening, which is the key trick here. So the mice are released for screening at similar ages, pedigree by pedigree, all at one time. And the genotypic data of all of the pedigrees, mouse by mouse, is uploaded to the computer and stored there. Whenever phenotypic data are uploaded to the computer by the screeners, who are students and postdocs in the lab, at that point the computer immediately calculates a linkage score for every mutation using dominant, additive, and recessive models. And this depends on a program that we've written that's called R-Link. It's a program written in R. And it's the computer rather than a human observer that calls a phenotype and that assigns linkage, as I'll show you. The data are then retrieved from a growing database that accumulates all of the mutation data and all of the screening data searchable according to a number of parameters, and that program is called Linkage Analysis 3.0. Let me show you how this works in practice. We can start with a very simple phenotype. It happened that we had a pedigree R0491 that had a total of 48 mice. 39 were designated unaffected and 9 were designated affected, as this was in the days when we were still naming phenotypes beforehand, and this was one of our early tests. We knew that there were 68 candidate mutations in this pedigree, and the question was, which one causes dwarfing of the mice? This pedigree was analyzed, as all of them are, for genotype using the ion torrent system after AmpliSeq is performed. These machines can analyze about 1 billion base pairs per 8 hours, but they're very nimble and they work on a chip-based system, so one can look quite quickly at different target exons to amplify. And the data come back in a tabular form from the ion torrent. You can see that each column here represents a different mouse, each row represents a different mutation site, and we're representing here the number of variant calls as we've sequenced these sites over the number of reference calls. Or, yes, that's correct. And the blue coding here represents then a homozygous variant. This would be a homozygous reference mouse, and this would be a heterozygote. Now, probably you can't look through the whole table to see how good the discrimination is, but we've pooled here 3 different pedigrees for you, and you can see that reference, heterozygote, and variant calls are unambiguously made when we do this, and we even black out areas that are between the peak points over a distance of about 20% or so. We're plotting the percentage variant calls on the x-axis, and in that way we make essentially no mistakes in genotypic assignment. So for this particular pedigree, although you can't read things, they're color-coded in the same way. We're plotting mice along the top, we're plotting mutations along the y-axis, and you can see that we've assigned genotypes to almost all of the mice. There are some cases where there are failures which are flagged, and this is the form in which one may review the data in the computer. We're able then to almost immediately see that there's a linkage peak using the additive model at the top or recessive model in the middle, and one even sees a very weak dominant effect of the mutation. And the mutation is quite unambiguously in KBTBD2, which is a gene about which nothing had been published previously. We now know quite a lot about this gene and what it does and how it works, and there are other effects that it has on the mice. But the point is that we found the mutation instantly. Now, this was in the old days when we were making our output in the form of PDF files. Later, we went to an HTML output format, and we had a few mice from this pedigree left, and we decided to do it in a strict way, not even looking at the mice, but merely weighing them and taking weight as a quantitative trait. And in this case, you can see that even with only 2 mice that are homozygotes, we get very strong linkage, plotting the log of probability of linkage along this axis and looking at all the mutations here. In this case, we don't discriminate between 2 closely linked genes, but usually we're able to do so. As of a few days ago, we had analyzed 34,525 allelic variants that fell into nearly 14,000 genes in the screening pipeline I showed you. And these mutations resided within a total of nearly 20,000 mice from 570 pedigrees. Now, it's interesting to think that those mutation sites were in all queried a total of 1.6 million times in all of the screening tests that we did, because of course you have many mice per pedigree and you do many screens on every one and you're testing many mutations for every mouse. That makes the process quite fast and efficient. We know that among the mutations were 2,612 null alleles that fell into 2,290 implicated genes. Not all of those were tested, of course. Not all of them were transmitted to homozygosity. Some of them were embryonic lethal, undoubtedly. But we know that 1,473 null alleles were tested at least once. 1,096 were tested at least twice. 803 were tested at least 3 times in the homozygous state. We take a single test of a homozygous mutation to be a good test for recessive phenotype, and we take 3 times to be a very stringent test for recessive phenotype. So we can say we've looked in a good way at about 6% of all genes, and in a very stringent way at about 3.4% of all genes, just when we're considering the null alleles and not any of the other alleles that we've generated. We have to have a console for looking at all of the data together, for retrieving data over time. And this is the Linkage Analysis 3.0 program. It's possible to look at simple Mendelian linkage, and as I'll show you also at complex trait linkage, using this switch here. One can do a wildcard search for genes or for screens, and one can use Boolean logic for that. One can look at specific mice or named phenotypes in our collection. One can restrict the search to different types of alleles or different probable outcomes of allelic variation. One can insist on viewing a mutation a certain number of times or more, 3 times or more, 5 times or more, whatever one wants. And the user sets the cutoff for significance. Almost always we apply Bonferroni correction in order to be confident about linkage. It's also possible and advisable to look for those things that score both with normalized and raw scores in the assay. That cuts down false positives a lot. And one can also specify the direction of change of a phenotype. There are other things that one can do as well, but in this particular case, we might be interested in looking at simply one screen, let's say something that affects CD8 cells, their number, their abundance, or their expression of certain markers. And here I've set the value for 3 observations as the minimum significance, and we've taken a p-value of 0.002. We have applied the rule of both raw and normal observations, and then we click Submit. What one gets back is a table in which one has a listing of genes, chromosome coordinates, the probable effect of the mutation, including the polyphen 2 score, where the mutation is a missense error. One also has a listing by pedigree, which is the usual way to display these things. One can see the screen name, the number of homozygous reference heterozygotes or variant homozygotes, and then most importantly, the p-value scores for the additive, recessive, and dominant mapping efforts. And wherever you see a red number, that's something that exceeds the probability threshold that you set. We find a lot of things that are known, like thymus, and we find things that are unknown, like SNRNP40, a component of the U5 spliceosome. We find that that scores very strongly in a test for the number of CD8 T cells and for the mean fluorescence intensity of CD44 expression by those cells. And if one simply clicks on one of the p-values immediately, you see the Manhattan plot. You may mouse over any of the mutations and see just what gene is represented by each point. And in this case, we see it's SNRNP40 that gives peak linkage score. We see what the linkage score is. If we then left-click, we immediately see quite a lot of information about that gene, including a link to MGI, of course. We see also the smart illustration of the protein after mutation. In this case, the mutation involves a splice acceptor site. One can click on the predicted gene model, and one sees what is expected of the mutation that removes exon 5, and there's a great deal of other information about the mutation also. If one right-clicks on the mutation, then a distribution of the phenotypic performance of homozygous variants, heterozygotes, and reference mice is displayed, and these are wild-type animals that are done in parallel when the screen is performed. One can also toggle between the normalized and raw data, and one sees that in this case they don't change particularly. We're looking here at elevated CD44 mean fluorescence intensity on the CD8 cells. Now, maybe you are not entirely convinced by that. One always has to look at a second allele to be confident about a phenotype. It's been our practice to use CRISPR knockouts to generate second alleles, but gradually this is becoming less and less important, and soon it will be a thing of the past and we won't need to do it anymore. Of course, already, having hit 14,000 genes, we've hit some genes many times, the larger ones sometimes dozens of times. And whenever this occurs, it's recognized by the computer, which incorporates the new allele into what we call a super pedigree. Eventually, all mutations will be incorporated into super pedigrees. They'll all coalesce, and with multiple alleles, there's growing confidence about the association between phenotype and genotype. So we might wonder whether a super pedigree already exists for this mutation, and we can click on this link here, super pedigrees, to look at the super pedigree analysis program. One can search globally with this, but in this case, we'll simply enter SNRNP40 as our search term, and we can look also for the CD8 responses, and we click submit. And then we see that indeed there are multiple alleles involved here. We see that there's one allele that's present in 13 different pedigrees, and there are 4 alleles that are represented in 16 pedigrees for this particular gene. We can look just quickly at the first one, and here we see quite a different picture. You see something of a blip. You see, first of all, an enormous log score. You have a probability value here of something close to 10 to the minus 28, favoring linkage. You see that there are many, many more genes, because in this case, in all of these 11 or 13 pedigrees, I don't know how many we have here, this mutation has been incorporated, and the other ones have been scattered, but the likelihood that they're involved in the phenotype is diminished increasingly. And so we can have great confidence that this is the causative mutation. We can, of course, right-click on this point, and then we see the performance of all of the mice from all of those pedigrees pooled together. We're looking at normalized raw data here, and I'd like to point out to you that this is the type of phenotype that would have been almost impossible to map if one had wanted to do so. You could never have done it by a qualitative approach because there's so much overlap with the heterozygous population and even with the wild type, and yet there's absolutely no doubt that the mutation has this phenotypic effect. Now finally, there are cases that are familiar to all of you who have done ENU mutagenesis where you have what looks like a reasonable phenotype, but it doesn't behave in a Mendelian fashion. It doesn't seem to transmit, or it transmits at far less than the expected level, or it transmits with variable penetrance, and usually you throw up your hands and you move on to something else. But we decided to be quite demanding about this and to consider that even when we make as few as 100 coding changes, sometimes we generate complex traits. And that appears to be the case. We made a heat map in which we look at phenotypic performance in an additive, recessive, or dominant fashion, combining all of the mutations with all of the mutations. And so we get a symmetrical kind of display, and one can mouse over it, and you see that this particular point seems to be the brightest in the heat map. And you may click on it, and there you see that indeed we do have additivity. We have a situation where there are 2 mutations, one in PIK3CD, which is a known gene with immunological consequences for the T-independent B-cell response, which is the assay here, and another in a gene that has no known involvement in that response. And if you have double heterozygotes, you have perhaps a small decline in performance compared to homozygous reference mice at both alleles. If you have a variable allele at homozygous variable allele at the PIK3CD locus, you have a decline in performance. But adding, even in heterozygous form, a mutation from RAL-GAPA1 decreases the response further, and double homozygosity is about the same. Our program will pick up this type of additive effect. It will also pick up inhibitory effects, which are equally common. It's kind of surprising, but it's not uncommon that one mutation will nullify the effect of another. We can project the destruction of the majority of genes and the analysis of their phenotypic consequences in this set of assays within about 3 years' time, which is something fairly remarkable to me. We don't have to wait for all the knockouts to be generated. Just in our lab, we're able to survey all of the genes in the genome, or nearly all of them, for phenotypic effect within a very short period of time. In fact, the bottleneck has shifted again, and I can say that the new bottleneck is simply the production of mutant mice. We're able to screen a bit faster than we're able to produce large pedigrees for this type of automated analysis. It's not only null alleles, of course, that we look at. We're intending to have a very deep allelic series at most loci. And we also see the prospect of understanding complex traits. The question might be, could we do this in humans? In the future, obviously, we're talking now about everyone having their genome sequenced at birth. What if everyone on the planet were sequenced, not only whole exome, but whole genome? Well, we know that all humans are related to all other humans. We know also that they're not really traceably related. There are only a few generations of us that survive on the planet, and the ancestry that links, let's say, a peasant farmer in France to a peasant farmer in China may be lost forever, is very likely lost forever. On the other hand, in the mouse, we started with a known ancestor, the C57 black 6 strain, and we introduced mutations piecemeal, bit by bit, and that is traceable. The humans sustain new mutations, of course, and these create phenotype. They're one source of phenotype. They also recombine old mutations, and it's possible to track down the new ones much faster than it's possible to track down the old. The power of the mouse now, and I think for a long time into the future, is that it's an excellent scout for human phenotype. It's a very common story that diseases are identified first in the mouse and then found to apply also in the human. And I think that's where JAX has absolutely a dominant power in coming to human genetics. This effort, of course, involved a lot of people. I have quite a large group now with about 55 people. The mutagenesis and production is done by Tiana Purrington in collaboration with all of these other people in the vivarium. Lindsay Scott is in charge of the exon sequencing and targeted genotyping. The screening for immune phenotypes is done by a collection of students and postdocs shown here. And we have a strong programming and informatics group that wrote the software I showed you. And finally, we have a pipeline for validation that's directed by Xiaoming Zhan. Thanks very much.